Hello everyone. I hope you are doing well from the last video that we made on the introduction to human anatomy and medical physiology. So today I will be introducing us into histology, which is a sub-branch of human anatomy. So as you can see there, you can see we have a cell that has various organelles, and then you can see a microscope. So histology basically is study of cells, study of tissues, and the use of microscopes to observe the tissues and the cells. So this is a brief overview of what we'll be looking at. So as we all know, in the cells, we have organelles such as mitochondria, the nucleus, nucleolus, and the rest. So here, the organelles make up the cells, and then the cells make up the tissue make up two or more tissues make up an organ, and then two or three organs, or maybe more than three, they make up an organ system, and then a couple of organ systems make an organism. So in histology, our discussion majorly will be on organelle, cells, and tissues in terms of their microscopic study. So we'll cover a few definitions, and for the few definitions, the first one that we are going to cover today is anatomy. And as you all know, anatomy is a study of structure of the body. So in your definition, you must have the word structure of body and its various parts. So for example, you want to study the digestive system, um, you're studying its structure, its makeup. That's anatomy now, and it's also the various parts of the digestive system. So starting from the esophagus, the stomach, the rectum, the jejunum, the ileum, that's anatomy now because you're studying the structure of the body. And of course, you know physiology. Now, after you've studied anatomy, the structure, you need to study the functions of that body part, of that structure that you've talked about. So, functions of the structure of the body and its various body processes, that's physiology. So, in a definition of the word physiology, you must have the word functions of the body and its various body processes. Then for histology now, this one should be a little bit simple because histology is an ideally study of tissues microscopically. So that's the reason why in the first slide we saw there is a microscope there because that is what histology is all about, studying the tissues microscopically. And then you can also have cytology. So cytology is basically study of cells. So logos is study, then cyto is cell. So study of cells is known as cytology. So, why do you need to study histology? Why do you need to know what histology is? So, why study histology? First of all, it will enable you to understand the body and cellular tissue levels. So, for example, you want to know what happens at a cell stage or what happens at a tissue level. So, for you to see what happens in terms of ATP formation, in terms of the structure of that tissue histologically. So, for example, we usually talk about the oral cavity, so the mouth. The mouth has parts, it has a mucosa, the epithelium itself. So, to understand the body at cellular and tissue levels, what type of cells form this tissue? That's the reason why we need to study histology. The other reason why we need to study histology is because you need to build a strong foundation for pathology. So, anatomy and physiology is a study of normal processes, while pathology is a study of abnormal processes. So if you know the normal, you'll also be at a very good point of knowing the abnormal, which is pathology. So building a good foundation for pathology, you have to study anatomy and physiology and cytology. You have to understand the normals that you go to the abnormal, which is pathology now. So once you build that foundation of pathology, it brings you to helping to know how to diagnose various diseases through tissue analysis. Of course, through histology, you learn the normal histology of various tissues. I know when you go to pathology, you learn the histology of the various abnormal tissues. So that's the reason why you can be able to diagnose that this person has maybe a cancer. This tissue has cancer, this tissue does not have cancer, or maybe the various stages of cancer, or maybe inflammation, such as things that you can be able to observe through the microscope histologically. Then it will train you to observe and think critically. So once you look at the microscopes, you'll be able to think like you look at the microscope, then you're thinking which tissue is this, or maybe what are the functions of the tissue that I'm looking at, or what are some of the maybe tissues that can have such a structure. So you'll be able to train observation, when I'm looking at this is the tissue that I'm looking at. And also it gives you the critical thinking skills. And just by the end of it all, by the end of learning histology and anatomy, you'll come to appreciate the beauty and complexity of human tissues and even the complexity of human beings. So starting from the small organelle, learning the function of that mitochondria, that Golgi apparatus, then you go to the whole cell, then you go to the tissue itself. Studying it microscopically just helps you to appreciate how various complex processes happen and how they can come up and provide maybe energy, provide some movements such as some movements and maybe things that maybe you didn't know about. So it will help you to appreciate the beauty and the complexity of human tissues. And now we have to discuss the structural organization of tissues. For structural organization of tissues, of course, I had alluded to it a little bit earlier, where we talk about the organelle, the cell, the tissue, and the organ. But we start from the small, tiny things that we studied in chemistry. So we start from an atom. So for example, you have another hydrogen atom. Then the maybe two hydrogen atoms come together and form hydrogen molecule. Then from the hydrogen molecule, you go to maybe something bigger. Maybe the hydrogen molecules, they come together and they give you macromolecules. Then the macromolecules, they form now the small structures that you see in cells, that is the various organelles. So for example, it's a mitochondria, you can have a Golgi apparatus, you can have ribosomes, you can have rough endoplasmic reticulum. So collection of a couple of organelles will give you a cell. Then various cell types will give you tissues. Then multiple tissue lineages will give you an organ. Then various organs, for example, now the esophagus, the stomach, the small intestine, the large intestine will give you the digestive system. So from organs, we get organ systems. Then from the organ system, you come across and various organ systems, usually about 12 to 13 of them. So those organ systems, they come up and give you an organism that's for us is the human beings. So maybe to just show you how the complexity of human beings are, is that, for example, the DNA that we usually have. So in a cell, for example, you can be able to appreciate that cell, it has a nucleus. And then in that nucleus, it has chromosomes. So those chromosomes are made up of telomeres, and those telomeres are made up of nucleosomes. So those nucleosomes, of course, will be formed by the DNA double helix that is being formed by purines and pyrimidines. So the base pairs are purine and pyrimidine, which are just very small adenine, guanine, cytosine, and diamine. So all those are small structures, compounds that enable formation of the human nucleus, the nuclear material. So that's the complexity of what we are talking about today. So we'll go to basic tissues, and in these basic tissues, We need to understand that there are just four of them. There are basically four. We have four basic tissues in all the organisms, basically all the organisms. You have four basic tissues. One of them is the epithelial tissue. The second one is a muscle tissue. The third one is the connective tissue, and then the nervous tissue. So for epithelial tissue, it lines various body cavities. For example, what you can see, be able to see now if you look at your skin, that's epithelium, that's epidermis. For the muscle tissue, of course, you know, you usually have skeletal muscles, cardiac muscles of the heart, and smooth muscles that are found in various organs. Then you have connective tissue. So connective tissue, just by that itself, it connects various structures. So for example, bone, you can have various cartilages connecting one part of the body, ligaments, all those are connective tissues. Then nervous tissue is one that now ideally enables communication. It's actually, it connects the various body parts through communication, and that integration comes from the nervous tissue. So for the muscle tissue, this one is quite simple for us. As we all know, we have three types of muscles. We have, let me start from here. We usually start with skeletal muscles. So skeletal muscles are actually all the muscles that you've seen, that you've ever seen actually on your body that enable the movement of your body. Those are skeletal muscles. And now histologically, we're supposed to know the characteristics of skeletal muscle. For example, you can see they are cylindrical in nature. Then this structure that you see that are somehow like yellow structures, those are nucleus, and they are multinucleated. So cylindrical, multinucleated. Then you can see they are somehow parallel to one another. Then you can appreciate the presence of striation. These things that looks like they're lines. Those are striations. And then there is no branching that is present in skeletal muscles. You go to now cardiac muscles. So cardiac muscles are found in the heart only. And you can see the difference between them is that for skeletal muscles, they are parallel to one another and there is no branching. But in cardiac muscles, there is branching, as you can see there. Then they are interconnected to one another. Of course, these ones will be uni or binucleated. So one cell will have one or two nucleus in it. Then also this one has striations, as we can see. So that's cardiac muscle. But we will study the histological organization of muscles in a later video. So feel free to look out to our YouTube channel and other YouTube channels around to see the various characteristics of muscle. Remember, it's an introductory video, so it shouldn't worry you much about the specific characteristics of specific muscles. We'll study them more once we do that. Then the last muscle type is a smooth muscle, which is found in various visceral organs. So it's found in almost each and every part of the digestive system, apart from maybe the esophagus. So from the middle third of the esophagus up to the proximal part of the anus, you have the skeletal muscles. Sorry, you have the smooth muscles. So those smooth muscles, they enable peristalsis, they enable major migratory complexes, those movements that happen every 90 minutes. It will enable now some processes such as churning that happen in the stomach. So all those movements that happen in the digestive system is due to smooth muscles. Also, not to forget that smooth muscles will also be found in the reproductive system. For example, now the fallopian tube, the uterus will find smooth muscles being present. The bladder also will contain smooth muscles. So that's some locations where you can have smooth muscles. So smooth muscles found in the various visceral organs. For the epithelial tissue, we'll go to epithelial tissue. So by definition, epithelial tissue is any type of tissue that covers body surfaces or lines cavities and maybe forms glands. So maybe you've heard about parotid gland, you've heard about thyroid gland. All those tissues that are found there are actually epithelium. So either epithelium that is lining the ducts or that is lining these epithelial cells are clustering around and are giving you the parenchyma of those glands. So that is epithelial tissue. So definition, you must have the word. It lines body surfaces, body cavities, and forms glands. That's simple definition. And for example, now we have the various types of epithelium. We can have simple squamous epithelium. So simple squamous epithelium means it's simple because it's one cell layer, and then it's squamous because squamous means flat. So simple, one layer, then squamous, they're flat. And then you can also see simple, why? Because it's one layer, then columnar, meaning the cells are somehow columnar in shape, as you can see there. Then you can have stratified. Stratified means more than one layer. So you can see, for example, now this one has more than one layer. So it's actually various. Then the uppermost layer is flat. So it's stratified squamous layer. Then simple cuboidal means it's just one layer by simple, then cuboidal. You can look the cells and even the nucleus itself looks circular. So if the nucleus looks circular, that's cuboidal in nature. If the nucleus looks oval in nature, that's columnar. So you can see the difference between simple cuboidal and simple columnar. Then you have stratified cuboidal. So stratified we say it's more than one layer. So you can see here, so one, two, those are two layers. That's the reason it's stratified. Then cuboidal, why? Because the nucleus is circular in nature. There. Then you go to transitional epithelium. So for transitional epithelium, this one is found in the urinary system, majorly the bladder, the urethra. You'll find them having transitional epithelium. So transitional means it can be able to transition. It changes in shape. So it's actually simple cuboidal in nature. Then after some time, it's certified squamous because of how it's stretched. So transitional means it's changing in nature. So transitional epithelium is known as urothelium because it's found in the urinary system. That's transitional epithelium. Then you go to stratified columnar epithelium. Stratified two layers, and then columnar, you can see the cells are of oval in nature. And then we talk about pseudo-stratified columnar now. That means all the cells are coming from the baseline, as you can see here, but they have different heights. And then also you can appreciate the nucleus are not on the same level. That's the reason why it's pseudo-stratified columnar epithelium. So all the set of epithelium are the ones that now will be found in the body surfaces, it will line various cavities, and they will form the various glands and ducts. So what are the characteristics of these epithelial tissues? So they are closely packed cells, as you can see here. So for example, they are closely packed cells with very minimal intercellular spaces. So they're closely packed, minimal intercellular spaces. Then they are vascular, so they have no blood vessels. For example, in this, we can't see any blood vessel. You can't see any blood vessel. So avascular means they are not vascularized. They lack blood vessels. Then they have high regenerative capacity. So that means they can be able to undergo the process of mitosis very fast. So they are highly regenerative. So they contain the basal cells for regeneration. Then they sit on a basement membrane. So each and every epithelium that you see here, it's actually seated on a basement membrane. So basement membrane, remember this epithelial tissue, they usually have polarity. We have an apical part, we have lateral part, and you have a basal part. So these are apical parts, this is a lateral part, and then you have a basal part. So that's the reason I say they are seated on basement membrane and they have polarity. So what are the functions of this epithelial tissue? One of them is that they enable protection. For example, now the skin, the protection property. The digestive system is lined by simple columnar epithelium that enables absorption. So actually you can see a simple columnar epithelium has microvilli for absorption. Then secretion, talk about now glands. Various glands, they secrete the various hormones. Then sensation, of course, the epithelium will contain nerve cells that will enable perception of various sensations. Then, of course, filtration, we'll talk about proximal convoluted tubule, distal convoluted tubule in the kidneys, which contain simple cuboidal epithelium that enables the process of filtration and even reabsorption. So we go to the connective tissue. In this connective tissue, its definition should be a little bit simple in terms that this connective tissue is a supportive tissue that connects. That's the word itself. So connective tissue connects or binds and protects other tissues and organs in the body. So it's either connecting or binding or protecting or supporting other tissues in the body. So that's a simple definition of the connective tissue from it. So what are the components of connective tissue? As you'll see, it will contain various cell types. For example, in this, you can see we have an adipocyte here. So the one that enables like formation of fat, adipocytes, stores fat, adipocytes. Will also contain macrophages. Of course, we know macrophages will have various names in various tissue locations. Then we'll have plasma cells. Of course, we'll have now some fibroblasts. So you'll have fibroblasts, you'll have macrophages, you'll have adipocytes, you'll have plasma cells and mast cells. So in this image, you're seeing mesenchymal cells, of course. You're seeing adipocytes, you're seeing macrophages, we are seeing fibroblasts. This is a fibroblast that you're talking about. And maybe other cell that you see is actually the mast cell. Then for the fibers, usually three types of fibers. We have collagen. So collagen is one of the tissues. For example, the one that you're seeing here, this one is collagen. And actually collagen provides the strength. Then elastic fiber now is one that now provides the elasticity, the recoil comes from elasticity, as you can see. So this one is elastic fiber here. So you've seen a collagen fiber, then this elastic fiber, the one that now enables the elasticity, the recoil property. And then the reticular fibers. Reticular fibers will be found in some organs such as the liver and the spleen. So the third component will be the ground substance. So this ground substance is like somewhat like a liquid or gel-like material that now will fill the spaces between cells and the fibers. So in between this collagen fiber and this fibroblast, in between you have the ground substance. Of course, now it will contain various components, various chemicals such as glycosaminoglycans, proteoglycans, all those will be found in the ground substance. So what are the functions of this connective tissue? So from the fibers, it provides support and structural framework to tissues. The other one is that it will enable protection of organs. The other function, given it has adipocytes, it will store fat. That is energy storage. And then it will contain some cells. For example, the macrophages, the mast cells, they have immune function. So they provide they act as immune defenses of the body. Then, of course, as you'll see, one of the connective tissue is blood that enables the function of the transportation of various compounds, maybe nutrients, maybe waste materials from the body. That's a function of blood, which is a specialized type of connective tissue. So, what are the various types of connective tissue? Of course, by this image, you can see you have adipose tissue, dense connective tissue. We have a bone as a specialized type of connective tissue. We have cartilage. We have blood as a special type of connective tissue and loose connective tissue. So broadly, they may be classified as loose connective tissue, dense connective tissue, and specialized connective tissue. So under specialized connective tissue, we have now cartilage that you can see here. Of course, we have three types of cartilages. We'll look at that later. or you can look that in our videos on the various types of cartilages. Then you have bone. Of course, bone is a specialized connective tissue, so you have a video on the same. And then you have blood as a third type of specialized connective tissue. Regarding now loose connective tissue, of course, we have areola, we have adipose tissue, and then you have reticular tissue. Those are loose connective tissue in terms of now the composition. So we call it loose connective tissue because it has less fibers and more of cells, while dense connective tissue will contain more fibers and less of cells. And this dense connective tissue, you'll find it in remember, you can have dense regular connective tissue that is found in tendons, ligaments, and aponeurosis, while irregular connective tissue, you'll find it in the dermis of the skin. So those are the three types of connective tissues. So we go to the third type of tissue. That type of basic tissue, which is the nervous tissue. Its definition, simple. It's actually a specialized tissue that receives, transmits, and processes electrical impulses throughout the body. That's a function of the nervous tissue. So it enables, it receives, it transmits, and processes electrical impulses throughout the body. So in the video on development or on the introduction to human anatomy, I explained it more on issues to do with the nervous tissue and the nervous system and neuroanatomy. So if you haven't watched that video, kindly feel free to go and watch that video. So the nervous system has various setups, but majorly you classify them broadly into two. You have neurons and you have neuroglial cells. So neurons, they're the ones that now transmits. Then neuroglial cells, they're the ones that now support the neurons. So neuroglial cells are actually supporting cells for neurons. Then neurons are the ones that do the function of receiving, transmitting, processing, and all that. So a neuron, for example, this is a neuron that you're looking at, it will have various parts. So it will have a cell body or a soma. So this cell body will have a nucleus in it, and then it will be having maybe a couple of dendrites on it. Then the cell body will be connected to this axonal terminal, uh, and then it will be covered by Schwann cells. These Schwann cells are the ones that produce the myelin sheath that you can see here. So it will have a couple of dendrites, cell body, then you'll have the axon, and then the axonal terminal there. You can see the Schwann cells wrapping around the axon. Of course, those are the various parts of a nervous tissue, of a neuron. Maybe you can go ahead and check on the various type of neurons. For the neuroglial cells, we talked about them. We can have astrocytes, oligodendrocytes, microglial cells, and Schwann cells. So for more info regarding the histological organization of the nervous tissue, feel free to visit our video on histological organization of the nervous system. Cerebrum, cerebellum, the spinal cord. We have listed all those cell types and you describe their characteristics. So what are the key characteristics of the nervous tissue? What are the key characteristics of a nervous tissue? It's highly excitable and it's conductive in nature. Highly excitable and conductive in nature. Then it's long and has branching processes. That is, the neurons are long and they have branching processes. And then these neurons are the ones that form the brain, they form the spinal cord, and they form also the peripheral nerves. So talk about the cranial nerves and the spinal nerves. All of them will be formed by neurons. And that's the nervous tissue, basically. Maybe now you might be wondering, what will you be looking at when you look at a neuron? For example, now this is a neuron, the cell body, and a couple of dendrites that you're looking at here. So this is soma, the cell body, and then you can see it has a nucleus in it, and then a couple of dendrites. So histologically, this is what you should be able to see when you look at the microscope. And then, of course, you can see this is an axon here. What are the functions of the nervous tissue? Just by the words, by the definition itself, it receives, so it has sensory input in it, then enables the process of integration and processing. So it integrates and processes the information that has been put into it. Then after integrating and processing it, it will give out a motor output. So it's received a sensory input, integrates and processes it, and then gives out a motor output. And then coordination of the body activity. So that's the simple functions of the nervous tissue from what you know. So for example, you put your hand in the fire. So the nervous tissue can be able to sense and send information to the nervous tissue. So that's sensory input to the brain. And then the brain now, which is a nervous tissue, integrates and processes it. And then it will give out a motor output through nerves, which are nervous tissue, um, to maybe the hand, and then tells the hand to contract a muscle, flex a muscle, extend a muscle to remove the muscle from, or the hand from the fire. So that's functions of that. And through that, it can be able to coordinate the body activities such as movements and such things. So we have a couple of tools for microscopy. So the simple one is a light microscope. Of course, you know that. We studied that in high school. Um, then the next one is electron microscope. So electron microscope, you can have two. Usually have TEMS. So TEMS is transmission electron microscopes, which are actually um, used for internal structures, used to study internal structures. Then you have scanning electron microscope, which is now SEMS. This one are used for surface details, so they can tell you the 3D of a certain structure. Then a microtome. This one enables the slicing of tissues into thin sections for tissue preparation of the slides, of the microscopic slides. So we have various types of microtomes. You can go ahead and look at the various types of microtomes, but remember this is introduction to histology. So I'm just covering the simple things. Then for digital microscopy now, this one you can be able to access it. Maybe you've connected it to your laptop, or maybe you've connected it to your phone. So once you look at images on the microscope, you can be able to send the images to your phone, or maybe to your laptop, or maybe you can even look at the images as you use the microscope to observe. So that's majorly about the various tools for microscope. But we'll also try to look at light microscope deeply. We need to know the functions of the light, the functions and the various parts of the light microscope. Welcome to the part that we were talking about regarding the light microscope. So the light microscope will have a number of parts. It will have the ocular lens that you can see here, also known as the eyepiece. You can see the objective lenses, of which have various magnification. You'll have the revolving nosepiece here, the arm, the stage, and then you have the clips. Then you can have the diaphragm and the condenser. This is a light source. Then this is a base of the microscope. Then you have the adjustment knobs. This is a fine adjustment knob. Then this is a coarse adjustment knob. So what are the functions? So we'll start with the eyepiece or the ocular lenses. So their function is to provide magnification of the image. So for them, they provide a magnification, maybe more because they are lens, they are just lenses. They provide magnification. of the image, and then it's also through them you can be able to look through the microscopes. Then you go to the body tube. So here you'll have the body tube. The body tube, we didn't mention about it, but it's the one that connects the eyepiece to these objective lenses. So the body tube connects the eyepiece to the objective lenses. Then you can have the nose piece, also known as the revolving nose piece or the rod or the turret. So these turrets, ideally it holds the objective. You can see where the objective lenses are. So they usually put on hold, put in place by the revolving nose piece. And also, if you want to maybe change the objective lens from one magnification to the other, you use the revolving nose piece. Then you have the objective lenses. So these ones, they provide magnification at various levels. Have magnification of times 4, times 10, times 40, times 100. So those are the various magnification according to how you want, how large the magnification you want it to be. Of course, you have the stage and the clips. So the stage is, you can see it's flat. So that's where you place the slide for observation. Then you have the clips, the stage clips which hold the slide in place and allow even the light to penetrate. Then we can talk about the fine adjustment knob and the coarse adjustment knob. So for the fine adjustment knob, it moves really the stage um, for smaller distances, and is one that now enables like precise image observation. For coarse adjustment knob, that one now moves the stage for some more larger distances for a rough overview of the object. Then, of course, you can see we have the arm. So the arm ideally supports the objective lenses, revolving nose piece, the body tube, and the eyepiece. You can see is where the arm. And then it's down that you can use it to carry the microscopes. Then talk about the base. Ideally, the base, it provides supports. So the bottom part of the microscope that provides stability and support the microscopes. Then you have the light source here. This light source, most of the books will refer it as illuminators. And this light source, as you can see, it projects the light upwards. So through it projects light upwards through the diaphragm. So you have the diaphragm here and the condenser. We'll talk about their functions in a few. So light from the light source through the diaphragm and condenser and through this slit here to the slide on the stage. And then through that you can be able to illuminate, so you can be able to see the structure that you want to see. Talk about the condenser and the diaphragm. So the condenser focuses the light on the specimen. So the condenser, by the way, it condenses the light to the specimen. Then the diaphragm, it controls the amount of light that reaches the specimen. So diaphragm regulates or controls the amount of light. Condenser condenses, it focuses light into the specimen. So that's majorly about the functions of the parts of the light microscope. So it's highly examinable. So just know the various parts, their functions that are important in the light microscope. So we'll talk about the common histological stains. Majorly we talk about 4 stains. And what is the purpose of, before you talk about the 4 histological stains, we need to talk about the functions of staining, or the goal or the purpose of staining. So you need to stain so that you can enhance the contrast. You can be able to look at specific structures well. So stains provide the contrast in the microscopic tissues. For example, there's a stain that can be able to stain the mitochondria in cells well than other cells. Others can be able to stain the nucleus well than other stains. So that's the reason you need to stain. And then through the staining, you can be able to observe their nucleus well more than a cell that hasn't been stained. So that's the purpose of staining. So one of the commonest stains that we use is known as hematoxylin and eosin, H&D. So this stain, it contains two parts from the word itself, hematoxylin and eosin. So hematoxylin ideally stains the nuclei. It will stain it blue or purple. Then the eosin part of this H&D stain, it will stain the cytoplasm and the extracellular component. It will stain it pink. And that's the reason why in a slide, this is what you can be able to see. So for example, this is a cross-section of the kidney, a transverse cross-section of the kidney. So what you can be able to see, you can see some blue or dark spots. So those are the nuclei of the cells. And then the other part that you can see staining there is actually the cytoplasm, and the extracellular matrix, the other is staining pink. So that is hematoxylin and eosin stain. The other stain we talk about the periodic acid sheaf, also known as PAS. So this periodic acid sheaf, it will highlight the carbohydrates. It stains the carbohydrates. So most of the times you used to check glycogen and glucose. Also, don't forget about mucin and the other types of carbohydrates, starch. So for it will stain some more dark, as you can see here. So the same cross-section of the kidney, but it's staining differently because it's staining dark, because you want to observe the carbohydrates that are present in that tissue. Other stains that we can have, remember, you can have Mason's trichrome. This Mason's trichrome stain uh, enables the differentiation between a muscle, a collagen, and even cytoplasm that is present in various tissues. So muscles will stain red, collagen will stain blue, the nuclei will stain thermal black in color. Remember also we have the silver stain. So the silver stain will enable the visualization of reticular fibers and nerve cells. To be specific, this silver stain, you will see it a lot will be staining the liver, staining the spleen. And then also you'll be staining the cerebellum, cerebrum, you'll be using the silver stain because it can be able to visualize the reticular fibers and the myelin sheath that is present in nerves. And then, of course, not to forget when you talk about now electron microscope, you talk about immunohistochemical stains, so which use antibodies to detect specific proteins in various tissues. So most of the times, these immunohistochemical stains are used in immunology labs. But the four most common stains that you'll see in histology labs, hematoxylin and eosin, a periodic acid sheaf, Mason's trichrome stain, silver stain, those are the most common stains. So why do you need to study histology? So remember we talked about histology being studying the normal microscopic organization of a tissue. And then pathology, it has histopathology, which is the study of abnormal part of tissue histologically. So the normal tissue, for example, this is a liver. You can appreciate how hepatocytes are cells of the liver. They're well lined together. They're actually forming lines, as you can see. These are hepatocytes. And then this is a podotriad. As you can see, it's well refined and has boundaries in it, as you can see there. While in a steatotic liver, one that has a lot of fat, maybe for a person who has been taking a lot of alcohol, steatotic liver, as you can see there. So the liver will lose its function. So histologically, what you can see first of all, the podotriad is disfigured. So this is a podotriad, disfigured. And then you can even start to appreciate a lot of fat tissue that are actually deposited in it. Unlike in normal liver, which doesn't have those fat deposits in it. So that's the reason why you can be able to identify that this person has steatotic liver by looking at that slide histologically. So there is no way you can know that is a steatotic liver if you don't know the normal histological organization of a liver. That's the reason why it's important to know first of all the normal histology of the liver. Then know when you study pathology, now you study the abnormal parts of the liver histologically. So that's normal versus pathological tissue. That's an example of what you're supposed to know. So how now will histology be assessed? It can be assessed using three ways. You can get MCQs on histology, you can get spot images on histology, and also you can get written essays on histology. So for example, we have a sample MCQ here, and the sample MCQ is here. So which of the following tissues, which of the following epithelium, sorry, which of the following tissues lines the inner surface of the blood vessel? So by the word lining, it tells you that we're talking about epithelium. So maybe you can pause the video and think about it, and maybe you can answer it in the chat box. But to be specific, um, the inner surface of blood vessels, as we'll see now in our video on epithelia, is actually lined by simple squamous epithelium. So simple squamous epithelium lines blood vessels. So these are histological organization of a blood vessel, most likely a vein. So you can see here we have this lining that you can see here. This lining, it's simple because it's one layer, and then this nucleus is flat. That's simple squamous epithelium of this blood vessel. And then what you can see here is actually blood, most likely the red blood cells. So the answer there is simple squamous epithelium. Then you can have sample short answered questions, SAQs. So maybe you can be told to describe the histological organization of the pancreas. So that's the pancreas if you look at it microscopically. So the pancreas, of course, you can see the one has been stained using HND. Why? Because you can see dark or maybe blue spots. That's hematoxylin, and then eosin giving it the pinkish part because of the cytoplasm and the extracellular matrix bit. So we haven't covered this well, but if you maybe we can talk about it. So it has two parts. The pancreas has two parts. It has an endocrine part and an exocrine part. But before that, histologically, it has a parenchyma and a stromal component. So parenchyma is actually the epithelium bits. The epithelium bits will contain now the endocrine and the exocrine parts, while the stroma is actually the covering of the pancreas. So stroma is actually the capsule that covers the pancreas, and then the various branches that the capsule sends in to the liver, to the pancreas to divide into various globules. So first thing first, the pancreas has a parenchyma and a stromal component. So remember the parenchyma is the exocrine and endocrine parts. Stromal component are the capsule that covers the whole of the pancreas and the branches that it has, dividing the pancreas to various lobules. Then remember you talked about the stromal component has a capsule, and then that same stroma divides the pancreas to lobules by connective tissue stroma. Then the parenchyma has exocrine and endocrine parts. So for example, in this image that you can see here, this slightly staining part is actually the endocrine part of the pancreas. It's usually known as islets of Langerhans. That's the islets of Langerhans, the lighter staining part. While the other part that you can see here is now the exocrine part of the pancreas. So we talk about the various pancreatic juices, pancreatic enzymes. They are produced by the exocrine part of the pancreas. The islets of Langerhans, the endocrine pancreas, now they usually produce hormones. So insulin that is produced by beta cells, alpha cells that produce glucagon, delta cells, we talk about somatostatin, the PP cells, the F cells will be produced by the islets of Langerhans. So we talked about the parenchyma, both exocrine and endocrine functions. So the exocrine part, which is this that we're talking about here, or what you can be able to see here, has various cell types. It will have serous acinine cells and central acinine cells, the one that now produce the enzymes. that same exocrine part of the pancreas has various ducts. So it can have intercalated ducts, it can have intralobular ducts, it can have interlobular ducts that transport the secretions to the duodenum. And actually a sample duct, you can see it here. So these are ducts. Most likely this one is a interlobular duct because it's very large. Then the endocrine part of the pancreas we talked about is known as islets of Langerhans, that is slightly staining. These islets of Langerhans will contain various cells, for example, alpha cells which secrete glucagon, a hormone, beta cells which secrete insulin, which is a hormone, delta cells which produces somatostatin, we'll have PP cells produce polypeptides, we'll have F cells which produce other types of hormones. And then maybe to wrap it up is that this pancreas has rich capillary network and a nervous supply. This has a nervous supply. That's a sample question on how you can have short answered questions asked someone in essay format for you to describe the histological organization of the pancreas. Maybe next time you can get histological organization of the liver, of the lungs, but don't worry, all these we have videos for them in terms of their histology. So feel free to reach out and watch the videos. And then sample image-based question, you can have such a thing. Of course, you haven't studied this, but we'll cover it. So the question will be, describe the histological organization of the structure displayed. So this one is image-based. You have to see the image and then identify the image and then talk about the histological organization. So for example, what is this? This is a thyroid gland. Why? Because of its structures. So you can see the colloid, you can see the follicular cells surrounding it, you can see the parafollicular cells. So this is characteristic of thyroid gland. So the structure shown is the histological organization of the thyroid gland. So why? Because you can be able to see the presence of thyroid follicles. So this is a thyroid follicle. And actually, thyroid follicle is lined by simple cuboidal epithelium. Remember, we talked about epithelium being found in glands. So simple cuboidal epithelium lines these follicular cells, and in it we have a colloid. And that's the reason why thyroid follicles are the functional units of a thyroid gland because they contain the follicular cells and the colloid. So as you'll see, this colloid actually stores thyroglobulin. We'll talk about it in a few minutes. So the follicular cells, which are CD skin cuboidal cells, will produce thyroxine. They are actually the thyroid hormones, thyroxine and triiodothyroid. That is T4 and T3. They are the ones that produce the thyroid hormones, the follicular cells. Well, the colloid now, this is a colloid that you're talking about, is the one that will store thyroglobulin. So this thyroglobulin is actually identified thyroglobulin, the one that now enables formation of thyroxine and triiodothyronine. So T3 and T4 respectively will be coming from thyroglobulin. And then we'll have these parafollicular cells here. They are also well seen here, and also they're also well seen here. So these parafollicular cells, also known as C cells, they secrete calcitonin, which is a hormone that regulates calcium levels in the body. So that is parafollicular cells secreting which hormone? Calcitonin, the one that regulates regulates calcium in your body. And of course, cytologically, like all the glands, as long as the gland is, all the glands will be containing rich capillary network. Of course, they'll also be containing nervous supply in it. And yeah, so what are some of the tips for you to study histology? What are the tips for you to be able to even identify the image that is on the background of that slide? To be specific, that's a slide of the parotid gland. But you'll be able to know more about it once you cover and practice histology more and more. So you study histology more and more, and study it well. If you attend histology and anatomy lectures, that's paramount. By using various virtual histology manuals, for example, histology guide, Michigan University, some of the universities usually contain virtual histology slides that you can be able to use. And actually, they have clear slides, you can be able to use them and study the various organs histologically. Then use revision questions every time after you're done with maybe a certain section of histology. You can use revision questions. You can use ChatGPT to generate revision questions for you in that part of the histology, and you can really do a lot in terms of your revision to histology. And of course, you need to review the content a lot. So regular reviewing of content that is just covered, and always and practice. Continue looking at those slides in the labs. Continue looking at those slides virtually. Remember, we want to develop that training, observation, and critical thinking. So you need to do that by practicing. So practice each and every time. And now, lastly, just to tell you that we have more prepared for you in terms of histology. So feel free to share the video to your friends, tell them to subscribe for more. We have a lot. We have a playlist for histology specifically that has videos talking about histological organization of the various systems, various parts. So feel free to subscribe, feel free to share with them with your friends. So I guess with that, I just want to wish you all the best in your academics and in your future career. In case of any questions, put in the chat box. And I just wish you all the best in your endeavors. Thank you.